Hi everyone, my name is Delaney James. This week's presentation is focusing on the urinary system, more specifically about the uptake of glucose and what the tubular maximum and renal threshold have to do with glucose's concentration within the blood and how that can affect diabetes and stuff like that. So, we are going to look at an overview of glucose. What is it? How does it normally get absorbed within the bloodstream? What is tubular maximum and its relationship to glucose, as well as renal thresholds uh, definition and relationship to glucose. We're going to look at how all of that ties into diabetes and what glucose's role is to that, because diabetes is very prevalent within our population as a country. And then we're going to look at some factors that actually can influence your renal threshold for glucose in a research project done by some scientists out in Asia. So, first off, what is glucose? Glucose, also known as blood sugar, is a monosaccharide, which is a singular sugar, and it is one of the body's main fuel sources. It actually comes from the Greek word for sweet, and glucose is mainly found in foods that are rich in carbs, like fibers, breads, fruits, things like that. Your cells use glucose for energy. It is broken down via cellular respiration to produce ATP. Hopefully, that is something familiar from your high school biology days. However, glucose cannot directly go into the cell, right? So our bodies do not just randomly take in glucose. All right, they first must enter the bloodstream, because glucose comes from the foods we eat. That glucose has to be entered into the bloodstream, and there it can be moved into the cell for it to be used. The hormone insulin is what's going to move glucose from the blood into the cell, and we're going to talk about that later once we get through renal threshold and tubular maximum and all that stuff. So here, if you can see my cursor moving, is the molecule for glucose. This is a pretty large molecule, so our cells cannot easily diffuse that through the membrane. We have to use insulin. And when we're done using insulin, the body needs to figure out a way to store it or get rid of it, or else our body is going to have way too much glucose. How glucose is absorbed. So, glucose is not normally present within the urine. Glucose is filtered throughout the body each and every day. And if it's not being used, it is stored as glycogen because it is a much needed molecule. So glucose is typically reabsorbed into the bloodstream by mechanisms that are located within the proximal tubule. So hence why glucose isn't present in the urine. It's being reabsorbed into our body. And if it's not being used, it's being stored as glycogen. So this whole reabsorption is done via secondary active transport, which is when specific symport carriers are going to transfer substances across the membrane. So this one in particular is a symport carrier that carries sodium and glucose. It is known as the sodium and glucose co-transporter. Very confusing name, I know. Transfers sodium cations and glucose at the same time. So from the lumen of the proximal tubule out here into the cell, going inward here, if you can see my cursor, out the lumen into the cell. So once the glucose is inside the lumen, inside the proximal tubule, glucose will diffuse down its concentration gradient into the plasma that is facilitated by the glucose transporter. So the whole goal is to get glucose into the cell out of the proximal tubule. So when we're talking about tubular maximum, we are referencing the maximum rate for reabsorption for any substance. That is what we refer to as the tubular maximum. So the amount of any substance that is filtered beyond its tubular maximum will escape the body within the urine. This limited rate is due to a limited number of carrier types within our tubular cells. And so, when every single possible carrier is saturated with a molecule, that maximum reabsorption rate is reached. So, when we're talking about glucose, we need to understand a few concentrations of glucose, how it's filtered, in order for us to understand our tubular maximum. So, the normal plasma concentration of glucose is 100 milligrams of glucose for every 100 milliliters of plasma. And because glucose is filtered freely at the glomerulus, it's going to pass through the Bowman's capsule at that same concentration. Um, and so we get approximately 125 milliliters of plasma being filtered every minute under normal conditions, healthy um, individuals with normal cardiac excuse me, rhythm. We're going to get 125 mils of plasma filtered. So the filtered load of glucose is 125 milligrams per minute. If you just do a little bit of math, that will lead us to the tubular maximum for glucose, which is approximately 375 milligrams per minute. Meaning, all right, we have 375 milligrams of glucose. That is what the carrier mechanism is capable of actively absorbing before our maximum transport capacity is reached. So we can carry up to 375 milligrams of glucose before we hit our capacity per minute. So with the normal plasma glucose concentration being around 100 milligrams per milliliter, the glucose being filtered every minute is easily absorbed because that's only roughly 125 milligrams, right? That is way less than our tubular maximum of 375. So this is what's going to actually keep our urine glucose free until the amount of glucose being filtered per minute is going to exceed the rate of reabsorption. Now I will have a graph on the next slide demonstrating that. But I wanted to mention renal threshold for glucose before that because they're on the same graph. So renal threshold is the plasma concentration that is reached when the tubular maximum for any substance, but in this case glucose, is reached, and that is when glucose will first appear in the urine. So this can be for any substance, like I just said, but the renal threshold for glucose is around 300 milligrams for every 100 milliliters, when the tubular maximum is 375 milligrams per minute. And the glucose filtration rate is 125 milliliters per minute. That's where we get our renal threshold of 300. So once our tubular maximum is reached, the rate of reabsorption stays constant at its maximum strength. So any increase in the filtered load, so any increase that we get in glucose that's being filtered, will lead to a direct increase in the concentration of glucose that is excreted within our urine. So, for example, if the plasma glucose concentration was 550 milligrams for every 100 milliliters, through some math, we would find out that the filtered load would be 687.5 milligrams per minute being filtered. However, our tubular maximum is only 375 milligrams per minute, and that's all that can be absorbed, leaving about 312.5 milligrams per minute spilling into the urine because we have surpassed that threshold that we can hold. So here's that graph I was talking about. If you can find my cursor over here, we can talk about it. So at a constant glucose filtration rate, right? If our concentration of glucose filtration is constant, the amount of glucose that is filtered per minute will be directly proportional to the plasma concentration of glucose. So here we have filtered per minute, right? That is directly proportional to the reabsorption rate. All the glucose that is present can be reabsorbed until we reach that tubular maximum, which is right here at about 375. And that is when we see this dotted line all the way across. This is all being reabsorbed because we have not crossed that tubular maximum yet. However, glucose is only filtered up to a certain amount. All right, we have to breach this maximum. Hence, we get the filtered concentration continuing up, increasing, and we see the amount that is being excreted increasing as well. These lines are parallel, so we see that the amount that is excreted is similar and on trend with the amount that is being filtered. So for every milligram that we increase of glucose, we get approximately the same amount in excreted glucose within our urine. So I mentioned that it's normally around 125 that is being filtered, so we're nowhere near that tubular maximum or renal threshold number. But why is this even important, right? It may seem like this is never going to happen because renal threshold is so high and average concentration is so low. However, glucose concentrations within plasma can be increased, usually caused by diabetes mellitus. This is an endocrine disorder where the body either produces too little insulin or it's producing enough insulin. However, our cells just are not responding to it. So they don't intake glucose very well. If that glucose is not intaking into the cell, it's going to be left within the plasma. When glucose remains within the plasma, because there's not enough insulin, the glucose concentration is obviously going to increase. Plasma concentration would increase the filtered load of glucose. However, only a certain amount can be reabsorbed due to the tubular maximum, meaning if the amount of glucose present surpasses the renal threshold, glucose will be found in the urine of those who suffer from diabetes due to the fact that insulin is not taking that glucose into the cell and it's remaining within the bloodstream. Diabetes can have an effect on your heart. It can have an effect on your kidney, your nervous system, and your eyes as well. I found that in my research. And usually this is because too much glucose in the bloodstream is damaging to those vessels that are carrying oxygenated blood to those parts of the body. So too much glucose is taking up that space for blood to carry vessels to carry oxygen-rich blood throughout the body. So this one study aimed to understand the factors that influence the renal threshold of glucose, specifically in patients with newly diagnosed type 2 diabetes mellitus. That is what 2DDM means, or adult onset diabetes. So researchers conducted a cross-sectional study on 1,009, very random number, hospitalized patients with 2DDM using stratified random sampling. Subjects had a myriad of tests done, so much I couldn't even list them on one slide, before even starting testing, and then they fasted for 12 hours. Blood samples were taken, and then subjects consumed 75 grams of glucose that was dissolved within 250 milliliters of water. And then after that was digested, blood samples were taken 30 minutes, 60 minutes, 120 minutes, and then 180 minutes after consumption, my bad. The blood glucose level was then monitored to obtain the mean blood glucose level, which was used to calculate the renal threshold of glucose. If you can find my cursor, that is what this first equation is showing, how they calculated that renal threshold of glucose. That is what RTG means over here. The researchers then analyzed the factors that influenced said renal threshold, and I'll go over some of those. They really analyzed a ton of factors. I'm only going to go over the ones that they highlighted as most applicable on the next slide. And they conducted multiple linear stepwise regression analyses to determine the main factors that did influence the renal threshold of glucose. I did want to point out that they did compare BMI, so that was calculated to determine the criteria for obesity, as well as the glomerular filtration rate, which was calculated. That is these two equations down here, one for male, one for female. And you can see that they used these numbers up here to calculate for the renal threshold as well. So this was just the setup for the experiment. Oh gosh, whoops. What they found was that the mean renal threshold for glucose of the patients was 203.58 milligrams per deciliter, plus or minus 55.22 milligrams per deciliter. They analyzed many variables to find a correlation, resulting in the following factors that influence the renal threshold. So here's that really long list I was talking about. So the factors that they analyzed include age, mean blood glucose, renal long neck, homeostatic model assessment for insulin resistance, homeostatic model assessment for beta cell function, levels of glycated hemoglobin, blood urea nitrogen, creatinine, uric acid, total cholesterol, low density lipoprotein, thyroid stimulating hormone, beta 2 microglobulin, and 24-hour urine glucose. Those were all the factors that they analyzed, and the results showed that there was no significant difference in gender, smoking or drinking history, waist to hip or waist to height ratio, which was used to determine BMI, and other obesity indexes and stuff like that. And systolic blood pressure. There was no significant difference between those factors and the mean renal threshold groups. There was, however, significant difference in age, course of disease, BMI, diastolic blood pressure, renal long neck, 24-hour urinary glucose, and 24-hour uric acid between the renal threshold groups. The linear stepwise regression showed that levels of glycated hemoglobin, 24-hour urine glucose, estimated glomerular filtration rate, D-dimer, and BMI should be included in the final model, with levels of glycated hemoglobin having the greatest impact on renal threshold, with 24-hour urine glucose following right behind, estimated glomerular filtration rate and D-dimer being kind of in the middle, and BMI being one of the least associated with renal threshold. So they revealed that renal threshold for glucose increased in most patients with newly diagnosed diabetes, and suggest beneficial anti-hyperglycemic effects, and the risks factors that were aforementioned, like age, course of the disease, BMI, and things like that. So here is a series of graphs that I plucked from the article. Here are the top 5 factors that they talked about. So here we have glycated hemoglobin. It shows a nice linear regression. This is why they chose that to have the greatest impact on renal threshold, with the least being BMI down here. And you kind of follow the order of 1, 2, 3, all the way in the top right hand corner, 4 down on the bottom left, and BMI down on the bottom middle, or 5, whatever number that is, with glycated hemoglobin having the greatest impact and BMI having the least amount of impact based on the linear regression of the renal threshold of glucose versus each of these factors. Here are my sources. I hope y'all learned something.